Hello everyone, welcome to all of you and a very hearty welcome to our today's speaker, Professor Raju Mishra sir. Sir has joined. We are very delighted to invite you here, sir. Thank you so much for giving us your precious time and joining us here in this FDP. I'm going to introduce you about sir. Dr. Raju Mishra sir is a professor in the Department of Computer Science and Engineering at IIT Patna, India. He obtained his PhD degree from IIT Kharagpur, MTech in Computer Science and Engineering from the IIT Bombay, and BE in Computer Science from MNIT Allahabad. His research interests span a design of distributed algorithms for mobile, ad hoc and sensor networks, cloud computing, big data, and wireless network. He has authored a textbook on cloud and distributed system really. He has offered online MOOC on NPTEL SWAM in cloud and distributed computing, big data computing. So welcome, sir. Please, sir. Hand it over to you, sir.
Okay. Am I audible? Can you
Yes,
all?
sir. Audible, sir.
Okay. Okay. Thank you so much. And the slides are visible in
Yes,
full
sir.
screen.
Visual, sir. But too large, sir. Too large. But okay, sir. Yes, sir. Yes, sir. Visual, sir. Fine.
Okay. Is it fine now?
Yeah, yeah. This is fine, sir. Perfect, sir.
Okay. Thank you. Okay. Let me first thank Dr. Dilip Kumar Chaudhary for for this invite, and because of this invitation, uh, I will be the presenter for this session. Mm. I will talk about the topic which is called as artificial intelligence in the drug discovery and the development. Primarily, the role of computer science nowadays is more into the drug discovery. Why? Because of COVID. You know that COVID requires a quick method of discovering the new drugs for the novel coronavirus. So the traditional methods were quite slow, and those kind of viruses, the medicines were tested, and all that method used to take a lot of time to develop the medicines. Now, the role of computer science will speed up, and this is the method which is required by our country also, and every other country to quickly apply the artificial intelligence in the drug discovery. So let us understand this particular topic in more details. So that will be the agenda. But before that, let me introduce myself and my group and the research which I do for the beneficial for all the faculty members, because this is a faculty development program, FDP. Now, as Dr. Dilip has already introduced me, so I am Dr. Rajiv Mishra. I work as professor in the Department of Computer Science and Engineering. Now, earlier I was working in the areas like distributed systems, cloud computing, the big data, sensor networks, IoT, and so on. These areas, you know, that now with the introduction of AI, machine learning, uh, of more use into the new fields like drug discovery and all, then we have taken the initiative during the COVID time that why don't we work in that domain, that is in the medical domain. So in the medical domain, what we found during the COVID is that how to find out how to discover the drugs for the novel corona viruses or new viruses which are evolving very fast. But the drugs are not discovered that fast, so that was the gap. And then there was an announcement came from Microsoft Research USA that they have invited the call for proposals, for research proposals in this direction, how the computer science and AI can be applied for this drug discovery. And we participated, we have submitted our proposal, and that got through. So my scholar and myself together pitched into this area because we were already there into the graph theory and AI and all that things. So we thought that, yeah, and then Microsoft gave their cloud on as a part of that research to conduct the research. So we did this kind of experiment in the Microsoft Azure AI and all that cloud resources which they have provided. So that is the basis, and that scholar is now completed its PhD, and that's some work regarding that we are going to present today with a little background. In the end, we will briefly describe about that work. And now that scholar has completed his PhD, and now he is offered as a postdoc in USA. So he is about to leave in the 7th of January. So that is what is the end of this entire business in this particular drug discovery. So now, if any of you is getting interested to know more details, so at the end I will give you the brief about our publications. So that publications will give you the complete description, a more insight if you want to start your research insight into that. Other than this, I work in big data, cloud, 5G, 6G, and so on. So if any one of you are interested for the research, that means if you are MTech and you are in the teaching business, then you are an ideal person who can contact us at IIT Patna to me. So what I will do is, if you want to be there in the job and do the PhD, then it is a part-time PhD possibilities are there. Or if you want to leave your job and come back and do the PhD, and after that build another career, going abroad and all that things, that also is a possibility. That means you have to come here through our projects. We will take your admission, and you can do the wonders in your career. So the people who are MTech and still in the job, for them it's a opportunity to contact me, and we can further help you to shape your career through the research. Okay, with that introduction, let us get started into this particular topic of today's, that is called artificial intelligence in drug discovery and development. So let us introduce some of the things that, as you know, that deep learning is neural network based intelligence learning, or it is inspired by the neurons, and therefore artificial neural network is used in these models. And so these models are used to train by the examples. So normally what you can see here is that how this can be applied into the field of medicine, because that medicine, if you want to apply these insights of deep neural networks or a deep learning method for rapid identification of potent, which is mentioned here in this particular slide. So this slide I'm not going to explain, but this slide tells you that you can apply various techniques into the deep learning, and using that technique you can conduct a lot of experiments without, or simulations of getting the drug identified quickly. So how that is all done, we are going to explain in the next slide, all that details. Then only we will come back to this to explain you more. So let us introduce here in this particular section, what do you mean by the field of medicine? So in the field of medicine, the drug discovery is the process by which the new candidate medications are discovered. This is very important background slide which you have to pay attention and understand this entire notion to get along with the other slides. So this is the basic background about the drug discovery. So drug discovery is a part of the field of medicine. Where the new candidate medicines are discovered. So new candidate medicine in so far those particular drugs are not being discovered and is not being prescribed by the doctors as a medicine. So why new medicines are required? Because novel coronavirus or new viruses which are appearing so fast, the process of drug discovery is not ready to do that. It requires new drugs to recover that. You may be knowing that for novel coronavirus, vaccines were designed, but there is no drug so far which can cure the COVID. So therefore, because this process is so slow that you require the new drug to be discovered. So for that, the new medications can go through. Now, this particular process of drug discovery is there to prevent the diseases and to treat the diseases. When you say that vaccine is not a medicine, it is just a booster, but it will not be able to prevent happening that particular disease. And how to treat the disease, you require a medicine that is in the form of a drug, and you have to discover the drug. So we will show you in the further slides how that is all done through the artificial intelligence and machine learning. Now, for the disease, a certain protein plays a crucial role. And for that particular protein, we have to find out a binding pocket or a binding sites. So now the new candidate molecules are generated that can fit into that binding pockets in order to cure the disease. So take this example. So think of that in a particular disease, a certain protein plays an important role. Okay, so now in that particular protein, let us say for that specific disease, you have to find out the binding pocket or a binding site, and you have to also identify from the candidate molecules which molecules can fit into this binding pocket in order to cure the diseases. So diseases will damage these proteins, and now you have to find out those candidate molecules which can be bind into that protein and therefore will heal this protein, heal this particular disease very fast. So this is what is to be understand. So you have to understand for every disease what is the target, what is the protein target, and then what are the candidate molecules that you have to search, and that molecules can bind into this particular protein target to cure the disease. So this is the process of finding the candidate molecules is called a drug discovery for a certain protein which plays an important role. So having understood this introduction, let us see in more detail about that. So here you can see that the drug is a small molecule that binds into the bio target, that is the proteins that I have explained in the previous slide, and modifies its functions to produce useful physiological or a mental effect. So once the small molecule binds this target protein, it modifies its function. Okay, and this modification of a function will also produce a physiological or different kind of effects to cure the disease. So whereas proteins are called large molecules, large biomolecules, which consist of the chains of amino acid residues. Here it is shown over here. It is the drug is a small molecule, but whereas the proteins are concerned, it's a large biomolecules which consist of the chains of amino acid residues. And the drug discovery, if you see, where you have the drug molecules, where small molecules for the drug, and you have a target protein. Uh, so what is the drug discovery? So drug discovery is the process through which these potential new medicines are being identified. It involves a wide range of scientific disciplines, including biology, chemistry, pharmacology, and so on. So we are not going in detail about that. It's an experimental research, but we are now talking only about use of computer science in that drug discovery. So as far as the rational drug discovery is concerned, it is the process of finding the new medications based on the knowledge of biological targets. So, so this is how when doctors can do, when the experts can do in the lab, why not? It can be applied and done through the simulation by the AI and artificial intelligence. So, the most commonly an organic small molecules that we have already told, and that is called drug discovery, that activates or inhibits the function of that particular protein, and that is what is called the drug discovery. And therefore you have to now you have to find out that molecule, small molecule which binds into that target proteins, and can inhibits the functions of the protein. And involves the design of molecules that are complementary in the shape and charge of a biomolecular target. Okay, so let us say that what is the drug discovery timeline normally? So traditionally, drug discovery process is time consuming and expensive. Why? Because you have a vast chemical space. So many molecules are there, comprising of, let us say, 10 raised to our 60 different molecules, which is more than that. So it's a huge space, and from that huge molecule space, you have to find out that particular molecule which can bind to your protein for functioning, uh, and making the treatment effective, and that is process called drug discovery. And selecting this particular molecule from a vast chemical space is a time consuming and expensive process. And the optimization of interdependent properties are also very much required in this particular process as far as the understanding is concerned. Now the AI, you know that artificial intelligence has the potential to accelerate the development timeline. So that is what that is why these researchers around the world, or the people who work in the field of medicines or in the drug discovery, they are now highly dependent upon artificial intelligence. So artificial intelligence has the potential to accelerate the development timeline by generating the molecules in the desired chemical space. So if the chemical space is too large, and you have to find out which is the potential molecule, so searching that large space is very efficiently done by artificial intelligence. So drug discovery and the development timeline, you can divide into these kind of stages, and these stages can be collapsed. So therefore, this drug discovery timeline, or you can discover the drug discovery very efficient. Now, what are the different approaches to look for the drug targets? So we are not going in detail about that. Only thing is that there is one more method called computer-aided studies, and there computer science or AI you can involve. So what is this computer-aided drug design? So computer-aided drug design, or called CADD, CAD, CADD, that is computer-aided drug design, deals with yeah, anyone who has unmuted. So as far as the computer aided drug design is concerned, it uses the structure based drug design, that is one way, or SBDD. The other method is called ligand based drug design, And here you can see that there is a drug which we have also shown in the previous slide. Uh, and there are two ways to carry the computer-aided drug design using ligand-based method is given over here. We are not going. The other important method is called structure-based drug design. Here, we have to identify that binding site, and then docking and scoring is to be done. As we have shown in the first slide, the introduction, the target protein and the docking means that particular binding of the protein or a small molecule into the protein or a bigger or a big molecule is called a docking, and then based on the scoring, that is which is more effective molecule for binding and making this particular treatment. So that scoring, or you can also think of a ranking, is to be evolved, and that then virtual is that process called virtual screening, and then you can find out that particular compound which is more effective in that particular disease, in the treatment of a disease. Okay, so this is the process computer-aided drug design I've explained. Let us go in more detail about that molecule representation. Now, when you say that it's a computer-aided drug design, CADD, then when you say computer, that means everything has to be represented in the form of the computer. And for example, the matrix you can represent in the form of a linked list. Sorry, a graph you can represent in the form of a linked list, or let us say that adjacency matrix. Similarly here, so what is the molecule? Molecule is similar molecular graph. Molecules are nothing but expressed with the help of molecular graph. So molecule representation, how you can do. So let us see what are the methods and how we can move forward. So for a molecule generation method, representation of the molecules are categorized in two classes. The first is called string-based method. So given a molecule, or the representation of a molecule like this, shown over here for caffeine compound. So you can generate a string-based approach. That means you can represent this entire molecular graph into the string, and that string is called CN1C equal NC2 equal C1C within the bracket equal O. And so this is a string. And this string is in a particular format which is called a smile string. So a smile string in the medical domain is quite popular. So given a molecular graph, you can convert into a string. That is called a string-based approach. The other method is called a graph-based approach. That means you have to now not go into that strings representation, but rather you have to protect the graph structure. So graph, here you can see that you have different atoms, and these atoms are connected in this molecule. So atoms means that you can represent in the form of the nodes, and the connection you can represent in the form of the edge. So this molecular graph will be now converted into the graph-based representation. So that molecular representation are divided into two classes. One is in the form of the string called smiley string. The other is called graph-based approach. Now, when you talk about the string-based approach, it uses that method of encoding into the sequence or in a string, and that sequence is called a smile sequence of the molecule, and this will become as an input to this particular computer-based drug design method. So either you can use the smile sequence, or if you are using the graph-based method, that includes the method that takes the molecular graph of the molecules as the input. So these are the two methods. So like you might have seen, the graphs can be represented using linked list or using the adjacency matrix. Similarly here, the molecules can be represented either in the form of the string called smiley strings, or in the form of the graph called molecular graph. That is what is represented over here. So in a computer-aided drug design, the molecules can be represented in these two types. So that is fine. So that is the basic understanding. Now the next thing is that These molecules are to be converted into the fingerprints. So fingerprints are nothing but, so for example, molecules is, if you think of a graph, then fingerprint is nothing but a vector, which is mostly discrete, and that can be that can be easily. So vectors are easily manipulable. You can easily manipulate through the computers, and not easy to reconstruct the graph from the fingerprints. So that is what is there. So from the graph, you can easily convert a vector, and from vector again back to a graph will be quite difficult, because many properties of three-dimension or multiple dimensions which is there into the graph capture will be collapsed into the vector. So that is what is called, from molecule you can convert into the fingerprints. So that is what is shown over here. You can have the discriminator or encoder network. So in this figure, you can see that you can apply the deep adversarial auto encoders for a new molecule development. Okay, and on the right side, you can see that you have convolutional networks on the graph for learning the molecule fingerprints. So that means to have this kind of convolutional networks on the graph for learning the molecular fingerprint. So there are various additional methods are available to deal with this convergence or representations of the molecule. So that is also categorized as the molecular representation. Now, another way is that from molecule to the string. So that is what we call it a SMILE. So the full form of the SMILE is simplified molecular input line entry system. SMILE is simplified molecule input line entry systems. So this kind of encoding that is ready for encoding and decoding with the sequential models, sequence to sequence learning, or this reinforcement learning and so on, can be easily applied once you convert the molecules into the string. But the strings, if you convert into the graph, they are not unique. So that means the graph, its uh, its uh, its structural properties will be lost if you convert into the strings, because strings, if you convert back to a graph, will not be that unique graph. So that means some properties will be lost here in this case. So lots of strings are invalid, and in the sense that although you have converted the molecules in the strings, but still they are not valid way of used in the computer-based drug design. So it has to be, this method is easy to work with the strings, because earlier days you can directly work with the strings, but now the artificial intelligence and neural networks are so powerful that the entire graph they can take as the input, and they can understand and process that. So, so again, let us see the molecular representation. So molecules, if you represent like a graph, here you can see that no regular or fixed structure, and the graphs are the permutation invariant. So number of permutations are exponential functions. The nodes, the probability of generating of a generated graph G need to be marginalized over all possible permutations. So multiple objectives. So diversity of the generated graphs, smoothness of latent space, and agreement with or optimization of multiple drug-like objectives. So these are multiple ways, multiple objectives are there. So if your objective of the computer design is not that complicated, then you can go for the string. But if it requires a very these multiple objectives, if it is required, for example, diversity of generated graphs, smoothness of latent space, agreement, multiple drug like objectives, that is all there, then you can go for this kind of molecule to the graph representation. And let us see the problem formulation now by understanding this molecule representation. So what are the problem formulation and objectives under this problem setting? So given a molecular data set Tg with the underlying data distribution Pg, that is for each graph Ax in the data set. So our aim is to learn how to obtain the new samples from the data distribution by employing the deep generative model. So this is the problem formulation objective is that main objective is to build the framework that can generate a highly potent active candidate molecules that can interact with the target protein structure with a high binding score. What are the commonly used data set in this drug discovery kind of or a computer-aided drug design? So these data sets are widely used with the deep molecular generation community. Zinc is a free database of commercially available drug-like compound for virtual screening. Then Chambl, CHEMBL, that is a large open access drug discovery database containing molecules and then GDB 13 and GDB 17, they are enumerated database of molecules of 13 or 17 heavy atoms of CNOS and so on, covering the drug-like range and including millions of isomers. So these are the data sets which you must know if you want to work in a computer-aided drug design, this as a field by applying the artificial intelligence and neural network. So let us see the single shot graph generation methods. So single shot graph generation is the idea of generating the graphs in a single step. In contrast to using iterative scheme to build the graph, this is similar to how the image generation works, where the images can be thought of as the regular graph. So here we summarize a few models that follow this scheme, Moi-Gann, Graph NVP, and so on. So this is a way that these methods are popular. Now let us explain them one by one through the research papers which is being published for single shot graph generation. Moi-Gann, which is the first model for this single shot graph generation. So Moi-Gann is published in a paper in 2018. Uh, the title of the paper is an implicit generative model for small molecular graphs. So they have named this particular method is called Moi-Gann. So Moi-Gann is an implicit likelihood free generative model for a small graph, for a small molecular graph that uses GAN, generative adversarial networks, on the graph structure data. So the authors in this paper use their model in reinforcement learning framework to generate the molecules with the specific desired properties. Although Moi-Gann generates nearly 100% valid molecules and is the first GAN-based model used for graph-based molecular design, it is susceptible to mode collapse, which results in a large number of repeats, and so on. So the next method which we have listed over here for a single shot graph generation is RVAE, that is constrained generation of semantically valid graphs via regularizing variational autoencoders. So that is being published in again 2018. So here the authors proposed the regularization framework for variational variational autoencoder VAs by introducing penalty terms for the output distribution of the decoder. Okay, and so we are not going in detail about that because these papers are already published, and if you are interested, you can refer them. That is available in the slide. Similarly, another method which we have expressed as graph and NVP. So graph NVP is the framework, is an invertible flow model for generating molecular graphs. So here the graph NVP is a model for the graph-based molecular generation using normalization, normalizing the flows, atypical invertible flow. Normalizing the flows are originally developed for image generation tasks. Okay, so here graph NVP method works by generating the graph in two steps. So first it generates an adjacency tensor array followed by generation of the node attributes X. So it does this by learning two latent representation, one for A and one for condition on A. So objectives of graph NVP is to learn an invertible model f theta with a learnable parameters theta, and the data set is used in this work is QM9 and Zinc 250K. So these papers are widely using because these are computer aided drug design. So they work along with the standard data sets. You see that most of these papers are using QM9 and Zinc 250K. So if you want to start the work, so you also have to now dependent upon these kind of standard data sets. And then these methods you can keep on changing or applying, maybe that you can improve upon this process of drug discovery. So MOFLOW, MOFLOW is also an invertible flow model for generating molecular graphs. So we are now giving you the research direction where from molecules you can convert or you can make a representation into the graph. So using graph NVP, like MOFLOW also works by generating the adjacency tensor A through a glow-based model, and then later on it generates and it uses the same data sets as used in the earlier paper. Now that was a single shot graph generation. Now let us see some of the methods which uses the iterative graph generation. So if the molecule is so complex that single shot graph generation will not capture the entire encoding, then you can go for the iterative graph generation method. So iterative graph generation methods, the molecules are generated in step-by-step manner by adding the components of the atoms to a incomplete molecular graph, one step of action at a time, that you know. So let us summarize the few models which uses the iterative graph generation method. NEVAE, DeepMind, JTVAE, and Graph Invent. These methods, let us explain few of them. So iterative graph generation method NEVAE was published again in 2018. And the title of the paper is Deep Generative Model for Molecular Graph. So NEVAE was the first model published to carry out the deep graph-based molecular generation and the architecture to molecular graphs whose encoder and decoder are specially designed to account for irregularity in the graphs and node order invariance and variable size. So again, the data set which is used here for this deep generative model for molecular graphs is QM9, 10K samples from Zinc. So you can see that the data set is almost same in different methods. So if you want to do a research or a study, you can also follow these papers, and like we have also done that to reach to our research. So DeepMind is a learning deep generative models for the graphs. So a team from DeepMind, you know that DeepMind is a research at the Google, used the GNN based model to generate the new graphs in an iterative manner. So we are not going in detail about that. Their models can generate both synthetic graphs and real molecular graphs unconditionally and condition the data. So the decision space is split into the three possible actions for building the graphs. So F node determines which whether to terminate the graph building process or add a new node to a graph, which determines whether to add the new edge to a newly created node and so on. which determines the score for each node, and thus determines which node to build the up the next, and so on. So that is what is, and also the data set which is used for this work is called Jing 250K, and 30K samples from Chamble. The next iterative graph generation method is JTVAE, that is called Junction Tree Variational Autoencoder for Molecular Graph Generation. So this work introduces the junction tree variational encoders that generates the molecular graph in the iterative steps. So data set is also used here is called Jing 250. Now the next is graph invent for method for iterative graph generation, and this is the learning deep generative models of the graph. So graph invent is a model for iteratively generating molecular graphs using GNN. It uses atomized atomistic level building blocks and has tiered TNN structure. So we are not going in detail about all these AI techniques, but we are only mentioning it because you can see that these are all the techniques, well studied techniques in the AI which is being used over here. Now coming up to our method of iterative graph generation, we have also done the contribution, and our contribution is generating the novel molecule for target protein called SARS-CoV-2 using drug target interaction based graph neural network. So this paper we have published. So Amit Ranjan is our PhD student that I have mentioned, and this is published in 2022 recently. This title of the paper is Generating Novel Molecule for Target Protein SARS-CoV-2 using drug target interaction based on graph neural network. So we have given this method how you can design a drug for coronavirus. So that was our aim, and finding out the drug is the drug discovery for that. So let us see what we have done. After this, we will open the floor for the questions. So what we have done is we have proposed a framework that takes account of both tertiary and sequential representation of the molecules and the proteins using a gated graph neural network, GGNN, knowledge graph, and early fusion network. So you can see that we have used several different methods to capture the molecule, the list of molecules which are to be selected as the candidate molecule and the target protein also. So you require different methods. So gated graph neural network, knowledge graph, and early fusion approach. Three approaches we have used for this molecules and the target proteins representation. Now then, these generated molecules from gated graph neural networks are screened using the knowledge graph. So knowledge graph will collect all the details, information about the interaction of the molecules, candidate molecules with that particular protein, and this information is kept into a graph called knowledge graph to reduce the search space. So so this knowledge graph will keep it, and then reduce the search space by discarding the non-binding molecules before being fed into the early fusion system. Now then, what we have done is binding the affinity score of the generated molecule is predicted using early fusion approach. Experimental result we have done, as I told you, the Microsoft Azure cloud we have used to do this experiment, and that experimental result shows that this particular framework which is being built generates the valid and the unique molecules with high accuracy while preserving the chemical properties. Now let us see the use of knowledge graphs. Claims that the entire generated data set of the molecules were reduced to roughly 96%, while retaining more than 85% of the good binding desirable molecules and rejecting more than 99% of molecules. So you can see that now when you reject 99% of molecules, so 1% of the molecule is a good number to go and to decide about the drugs here in this case. That is what is the point of research which we have been concluded. Now with this, let us stop at this point and let us open the floor for the questions. So if you have any questions, you can ask on this particular topic.
Okay. So participants, please, if some queries, you may discuss with sir. Please.
Hi sir, this is Rajesh. You have told that you have Azure platform to build this approach, a medical generation approach. So, can you explain more? What are the data sets you have used and how we will bind that one to generate this GNN graph?
Okay, so the data sets, as I told you that we have used the same data sets like in the previous method which we have shown you, that QM9 and Zinc data sets we have used, and these data sets are available because these are the public data sets on which many papers are being published, and GitHub repository is also available. The only thing is that you have to build a model. For that you require your own computational power or GPUs and all that things. For that we have used this one, Azure machine learning libraries. So Azure machine learning libraries, ML libraries, they have this kind of capabilities to build these graph neural network. Uh, Gated graph neural network we have built over the Azure, Microsoft Azure, and the experiments were conducted after that. So I think Azure is easier way to conduct the research. Otherwise, you know that what happens is that the data set is so large that if you download at your desktop, then you will not be able to download the entire data set because the data sets are so huge or so big. But if you use the cloud as the repository, so there not only you will be able to store, you have a lot of storage, but also you can use the number of GPUs whatever is very much needed here for graph neural network computations. So that is what the scheme which we have, since Microsoft has given us all that resources. So our students and Microsoft team also has helped him how to use that for building this kind of models.
Okay, sir. Thank you for that info.
So is there any more question? Anyone? So, okay, thank you so much, respected sir, for your valuable time. Your expert talk has provided us with invaluable insight about how deep learning models along with biological technology are used in rapid drug discovery. You also talked about computer-aided drug design and how molecular representation can be converted to a graph. You also informed about MOIGN for single-shot graph generation and about SARS COV to target protein generation using graph neural network. I'm sure our participants have attained a better understanding about all these topics. Thank you again, sir, for taking out your valuable time to impart us with your knowledge. Thank you, sir.
Thank you so much. Bye.
So, okay, uh, today's session is going to be over. We will join by tomorrow. Thank you.
Sir, I have a question.
हाँ बोलिए पूछिए.
Sir, We 15 to 16 students are from agriculture background.
एग्रीकल्चर बैकग्राउंड का हम टच आपको ऑफलाइन मोड में दिलवाएंगे, बोले हैं। ऑनलाइन में क्या है कि हमने रखा था, और एग्रीकल्चर का फिर आप लोग थोड़ा ज्यादा ज्वाइन किए। ये भी जितना दे रहे हैं आप देखिए, एआई मशीन लर्निंग पर दे रहे हैं सर लोग। और यहाँ पर आपको हमने रिक्वेस्ट किया है, एक दो लोग 100 किए थे वो डिनाई, एक्चुअली बहुत सारे लोग डिनाई कर चुके, यूएसए वगैरह का पूरा सडन बन गया, आज फिर है, ठीक है। लेकिन फिर भी हमने यहाँ पर इंटरनल भी बोला, रिक्वेस्ट किया कि नहीं थोड़ा एग्रीकल्चर का हमको लोग को देना है, और कुछ लोग एग्रीकल्चर का देंगे। अरे जितना टॉक है ये है, केवल डेटा सेट अलग होगा, फिर भी आपको यहाँ कुछ होगा। और 3-4 सेशन के लिए हमने और बात किया था, तो उनके घर में कुछ प्रॉब्लम हो गया, क्या कहा जाए, जिसको जिसको एग्रीकल्चर पर रखा था हमने रिक्वेस्ट किया था लोगों से, तो कुछ कुछ प्रॉब्लम, फिर भी यहाँ आपको मिलेगा, आप आइए यहाँ पर, आपको समझ आप देखेंगे कि हाँ, मेन चीज है कि आपको टेक्नोलॉजी जो भी है, एमएल एल्गोरिथम कैसे यूज करना है, ये जाए इस इंस्टीट्यूट से आपको, हमको लग रहा है ये उम्मीद आपका भी उम्मीद यही होगा। डेटा सेट वगैरह कलेक्ट करना, उसका स्पेशलाइजेशन आपसे बेटर हम नहीं बता सकते, बट फिर भी यहाँ के जैसे आपका एक प्रोजेक्ट हमारे इंस्टीट्यूट, आपके इंस्टीट्यूट में एमओयू हुआ है, जिसमें आपके संदीप राज सर काम कर रहे हैं डायरेक्टर सर के साथ, जो आया है कंसल्टेंसी प्रोजेक्ट, उसमें भी वो आपको हमने उनसे बोला है कि नहीं, हम लोग को अच्छे से एक्सप्लोर करना है, तो लोग करेंगे। आप आइए, हैंड्स ऑन सेशन है ना आपका, 5 दिन, पहला दिन इंडस्ट्री विजिट वगैरह भी है। सो ओके, थैंक यू। और
थैंक यू।
आप देखिए ध्यान से, आप लोग भी क्या है, बहुत सारे एक्सपर्ट टॉक वगैरह अटेंड किए हैं, सब चीज किए हैं, तो आपको मैं जरूर मानूंगा थोड़ा एग्रीकल्चर चीजें नहीं आ रही, मेडिकल आ रहा है, हेल्थ केयर एंड एग्रीकल्चर, ठीक है, बट डेटा सेट अलग है ना, टेक्निक्स तो वही सब लगाना है, वही करना है। उसमें भी हमने फिर से हमने बोला संदीप सर को कि नहीं, एक टॉक आप उस पर लाइए, तो आइए आप लोग देखिए, है ना। ओके हर्ष,
थैंक
सो
यू सर।
थैंक यू, यू ऑल may leave it, थैंक यू।